So, good morning everyone. How are all of you? So, we have to start with cell wall today up to where we discussed in the last class. Can you pin? Good morning, good morning, good morning. So, up to where we discussed, can you anyone pin the last topic? Good morning, good morning. Please say up to where we discussed in the last class. Prokaryotic cell already started, na? Done, finish also probably. Up to where we done? Prokaryotic details you have to say. Prokaryotic details. Shape of bacterial cell completed. Okay, have a look now. Have a look. Give your attention. Prokaryotic cell. So if we talk about prokaryotic cell, it include all the members of kingdom Monera. Right now, I think these are the area I told. Shape of bacterial shape also completed. Right. Now we are going to talk about a structure of bacterial cell or prokaryotic cell. A structure of prokaryotic cell. A structure of prokaryotic cell. Give your attention. So I am drawing the structure of prokaryotic cell. So if you observe a typical prokaryotic cell, their cell envelope is trilamellar. They have a which type of cell envelope? Trilamellar cell envelope are there. Cell envelopes covering of the cell is made up of 3 layers, and that is the reason I am using the word trilamellar cell envelope. So the outermost layer of the cell envelope is called glycocalyx. Second from outside is called cell wall. And the innermost layer of the cell envelope is called mesosome. Sorry, mesosome I am saying, plasma membrane. Inner membrane is called, innermost layer is called plasma membrane. Plasma membrane. Plasma membrane. All 3 together, this plus this plus this is called what? Cell envelope. You got the idea? So I am telling cell envelope of the prokaryotic cell is trilamellar, made up of 3 layers. Clear in the mind? So all 3 layers are fused together. It's not like they are separately present. They fuse together. Glycocalyx, cell wall, cell membrane fuse together and making a structure that is called cell envelope, and this cell envelope is trilamellar. Trilamellar. lamellar. Is it done? First I am drawing the structure, then I will explain everything, don't worry. So now I am telling inside the plasma membrane who is present? Cytoplasm is present. So I am writing here it is what? Cytoplasm. Cytoplasm. Cytoplasm is a universal structure. It is present in both prokaryotic cell and eukaryotic cell. Getting the idea or not? And inside the cytoplasm who is present, let me tell you, inside the cytoplasm you will find there is single circular genetic material is present, and that is called what? Basically nucleoid. Nucleoid. So nucleoid is present. It is what basically? Nucleoid is genomic DNA. Genomic DNA. Understanding the idea? It may possible in some prokaryotic cell, not in all, extra genetic material is present. And these extra genetic material is called plasmid. These extra genetic material is called plasmid. These are present inside the prokaryotic cell cytoplasm. Beside of all these, in prokaryotic cell, what you will find? Polysome is present. Who is present? Polysome is present. So what happened in polysome? Suppose that this is a mRNA. And with this mRNA, many 70S ribosome join here. So this structure is called what? Basically polysome. Polysome, or it is also called polyribosome. Polyribosome. Understanding the idea? So these are also present in which cell? A prokaryotic cell. Beside of that things present in the cytoplasm, inside the prokaryotic cell, you will find many storage granules are there. And these storage granules are called inclusion bodies. Inclusion bodies are there. It may be some of the ribosome present in free stage. It is not necessary a ribosome always making a polysome. So it is, you can say, about free ribosome. So I hope you got the idea what is present inside the cytoplasm of a prokaryotic cell. Picture came or not came in the mind? What are the things which is present? You have to say कि nucleoid is present, plasmid may present, polyribosome is present, inclusion bodies are present. So these are the structures present inside the cytoplasm of which cell? Prokaryotic cell. Now I am going to tell what additional thing present in the prokaryotic cell structure. So let me tell you, you may find in prokaryotic cell filamentous, long filamentous structure, that is who is present? Flagella is present. Who is present? Flagella is present. It may possible a tubular outgrowth, Pili is present. And you may find who is present? Small bristle-like structure present all over the surface of the body, and these bristle-like structure which is present all over the surface of body is called fimbri is present. So three kind of extensions are present. Got the idea? And these extensions are what basically? These extensions are flagella, Pili and fimbri. Now we are going to decode each and every one by one. Then who will finish? Prokaryotic cell will finish. Structure is clear in the mind or not? Tell me. So these are a typical structure of a prokaryotic cell. What the idea? What is the function of cell envelope? In one line, if anybody asks, cell envelope, what is the function? Protective in function. What is the function of cell envelope? protection. Understanding the idea? Is it clear? Now we are going to talk about each and every component. So I am starting the story with which one? Outermost layer, glycocalyx, then we will talk about cell wall, then I will say about plasma membrane. So have a look. I am going to say you about which one? Glycocalyx first of all. So if we talk about glycocalyx, it is outermost layer of cell envelope. It is outermost layer of which one? Cell envelope, and these glycocalyx may present as which one? Loose sheath-like structure. May present as loose sheath. Getting the things or not? And when it is present as loose sheath, it is called slime layer. It is called slime layer. And when these glycocalyx is tough and thick, it is called capsule. It is called capsule. So when capsule is present in bacteria, it is called capsulated bacteria. Getting the things or not? So what is the functions of the glycocalyx? Function of the glycocalyx is, one function is what? Protection. And another function is desiccation. Desiccation means what? It prevents the bacteria from which one? Drying, getting dried. Understanding the idea? Is it clear in the mind? Is it clear? Is it all right? So these are about the first layer, that is called glycocalyx. Nothing wrote more in NCERT, hence I am not talking much. Otherwise, these capsules have also a role in which one? Defense of the bacteria against phagocytosis. But then who will interfere? Immunology will interfere. That's the reason I'm not talking much detailing. NCERT wrote that much information, so it is okay. Now we are moving ahead. After glycocalyx, we are going to talk about the cell wall. So cell wall is what? Thick layer, provide shape to bacterial cell. Bacterial cell. If you talk about another things, it protect the bacteria. Bacteria is what? Prokaryotic cell from bursting. From bursting. Getting the things or not? And if you talk about the cell wall, it is made up of peptidoglycan. It is made up of which one? Peptidoglycan. Understanding the idea? So this is about the cell wall. You have to keep in mind, it is a thick layer and it provides shape to the bacterial cell. It protects the bacteria. Getting the things? Protective layer, and from which one? Protect the bacteria from bursting. Able to understand? And overall, any of the envelope is protective in function. That is a common one. So it is made up of which one? Peptidoglycan. If you talk more about peptidoglycan, these peptidoglycan is a polysaccharide made up of cross-linking of NAG and NAM. NAG is N-acetyl glucosamine, NAM is N-acetyl muramic acid. What the idea? Now, if you read NCERT writing, based on staining, there are two type of bacteria is there. Based on a staining of cell envelope, there are two type of bacteria is there. One is called gram-positive bacteria and another is which one? Gram-negative bacteria. gram-negative bacteria. Getting the idea or not? So the staining, why we are telling gram-positive and gram-negative? Because staining technique of bacteria is discovered by Christian Gram. Discovered by Christian Gram. C Gram in short I am writing. Christian Gram. And based on honor of his name, the stain is also called Gram stain. So he used crystal violet. It is a name of a dye. It is a name of a stain. It is what basically a stain, color. Crystal violet to stain means color, the bacteria. So the bacteria who retain the stain, gram-positive bacteria retain the stain. It is called gram-positive. Why is it gram-positive? Because Gram was the name of scientist who discovered it. So it is called gram-positive bacteria. And bacteria who not retain the stain, is gram-negative bacteria. And I am closing it here only. Of course, I can understand many questions may arising in your mind, like why one retain stain, how to do staining, which color will come, what is the meaning of retain a stain and not retain a stain. After that, what is the difference between gram-positive and gram-negative? Many questions may arising in your mind, but we are not going to give the answer of this question because it is not the need for the examination. NCERT not wrote about all these things. So when we earlier teach, at least we talk about more than half an hour on which one only, around 1 hour on gram staining. Method of staining, procedure of staining, how gram-positive, gram-negative differ from each other, why one retain the stain, why one not retain the stain. But now it is not the demand of the exam, and that is the reason we are just setting it here only. Getting the things, gram-positive and gram-negative. Based on which one? Staining technique, there are two types of bacteria there. So now we are going to talk about plasma membrane. Third layer of the envelope, plasma membrane. And if we talk about plasma membrane, so it is inner most layer of envelope, and this is thin and elastic. It is selectively permeable. Do you know the meaning of selectively permeable? It allows solvent plus selected solute to pass through it, and you can say like it is like eukaryotic membrane. It is like eukaryotic membrane. Means cell membrane of prokaryote and eukaryote at your level, NCERT wrote what? It is same. It is same. Both have a similar structure. So that is the reason I wrote it is like eukaryotic membrane. And I am writing one line. It interact with outer environment. Now you will ask a question, na? Ki, sir, it is the innermost layer. Outside of it, there are two layers. Outside of it, how many layers is there? Two layers is there. Look, na, cell wall is there. Glycocalyx is there. Then why plasma membrane interacting with the outer wall? Anybody can answer this question. NCERT also wrote plasma membrane interact with the outer environment. So why? A question arises na ki it is the innermost layer who are outermost, they have to interact. Now why plasma membrane is interacting with outer environment? Anybody can answer this question? Anybody can answer this question, why plasma membrane interact with the outer environment? Please try to answer. Pin your answer if you know it. Let me answer. See, we don't have that much time to, so let me answer. Because of, yeah, one right, very beautiful answer. Because of its selective permeable nature. Out of these three membranes, only plasma membrane is the living membrane. Who is dead? Cell wall and this capsule is not living structure. It is selective permeable membrane. Cell wall is permeable, glycocalyx is permeable. They are not stopping anyone to come. Who is coming, who is going, they are not caring. Who is caring? Plasma membrane. Plasma membrane are saying, oh bhaiya, why you are going in? Who are you? Have you permission or not? Show it. Then I will allow. If anyone want to go out, why you are going out? What is the reason? Getting the idea? So these kind of interaction, either anything coming from the cell, coming in the cell and going out of the cell is decided by that questioning done by whom? Plasma membrane. Who is not doing anything? Cell wall and cell, which one? Glycocalyx. And that is the reason I am telling, NCERT also wrote, it interact with the which one? Outer environment. Picture came or not came? Now we are going to talk about, so detail about plasma membrane structure we will study in which one? Eukaryotic cell. Meanwhile, I am telling you about enfolding, so extension of plasma membrane. Extension of plasma membrane. So look what are present as a extension of plasma membrane. There are two extension of plasma membrane is there. One extension is chromatophore. I have also shown it in the diagram. So if we talk about chromatophore, it is enfolding of plasma membrane which contain pigment. Which contain pigments. So these chromatophore is analogous to chloroplast. Otherwise you see na. So these chromatophore present in photosynthetic bacteria. Present in which bacteria? Photosynthetic bacteria. Now you please try to understand. In prokaryotic cell, who is not present? Chloroplast is not present, and you better know photosynthesis occur in chloroplast, and in prokaryote, chloroplast is missing, not there. So, whenever a prokaryotic cell doing photosynthesis, something must be there in them, na, who work like chloroplast. Something must be present in them, na, who work like whom? Chloroplast. Understanding the idea or not? Clarity came in the mind or not? Something must be there who work like chloroplast. Is it all right? Is it clear? And that structure is what basically chromatophore. So in photosynthetic bacteria, what is the job of chloroplast is there in eukaryotic cell? Same job performed by which one? Chromatophore. And hence, I have written here a line, it is analogous. A spelling error is there. Analogous to which one? Chloroplast. Analogous means what? Functionally similar to which one? Chloroplast. So it is present in which bacteria? All bacteria? No need. It is present only in which bacteria? Photosynthetic bacteria. So you got the idea about the chromatophore, right? It is present in which bacteria? Photosynthetic bacteria. And who is photosynthetic bacteria? Cyanobacteria. That is blue-green algae, iron sulfur bacteria, green sulfur bacteria. These are some of the example of which one? Photosynthetic bacteria, where who is present? Chromatophore is present. Now I am going to talk about mesosome. So mesosome, it is also an enfolding of plasma membrane present as vesicle, tubules or lamella. Got the idea or not? Mesosome present as what? Vesicle, tubules or lamella. Right. Now, what are the function performed by this mesosome? And it is very important. In entire prokaryotic cell, you don't forget, there is maximum chances, if a paper setter told to frame one question, they will prefer to pick from where? Mesosome. So mesosome is very important, you don't ignore it. So mesosome, first function I am writing, DNA replication and its distribution in daughter cell. So one of the function of mesosome is this. Second function of mesosome is cell wall formation. What is the second function? Cell wall formation is the second function of the mesosome. The third function of mesosome is increase surface area for cellular respiration and enzymatic action and for enzyme. Got the idea? So any enfolded structure, there is a thumb rule. I am making a pipe like this and I am making a pipe like this. You are finding both have same length, but which pipe have more surface area? Who are enfolded? So any enfolded structure have what basically? More surface area. Another function is what basically? Cellular respiration. This is another function of which one? Mesosome. Cellular respiration means it is analogous to which cell organelle? Mitochondria. So how interesting, in prokaryotic cell who is missing? Plastid is missing, chloroplast is missing, mitochondria is missing, and function of both chloroplast and mitochondria done by what? Extension of plasma membrane. So extension of plasma membrane working as chloroplast. So work of chloroplast is done by extension of plasma membrane. Work of mitochondria is done by extension of plasma membrane. Otherwise, you know na, in cellular respiration, link reaction, Krebs cycle, ETS, these all require what? Mitochondria. But in mitochondria is missing in which one? Mitochondria is missing in prokaryote, but beside of that aerobic respiration occur na? In prokaryote also aerobic respiration occur or not occur? Tell me, occur. So who is doing the job of mitochondria here? Mesosome. And that's the reason I wrote here, it is analogous to which one? Mitochondria. Another function performed by the mesosome is which one? Cell secretion. cell secretion. So these are the some of the function associated with which one? Mesozoa. Understanding the idea? So it is done. Is it clear in the mind? So who is over? Membrane is over. All three cell envelope is over. Clear? Now we are going to talk about what is present inside the cell. And the very first we will talk about which one? Nucleoid. And I will not talk much here because I already told about which one? Prokaryotic cell. Difference between prokaryotic and so nucleoid, it is main genome, main genetic material. And you know about it. It is single, circular, double-stranded, necked structure present in cytoplasm. I wrote about it earlier also. Got the idea? So present in cytoplasm. So nucleoid is over. Nothing more discussion. Now I am telling you about the plasmid. Plasmid. You have to know. This plasmid is what basically? It is extra genetic material. It is extra genetic material. It is not the main genetic material. It is extra genetic material. DNA, a plasmid is not called genomic DNA. Nucleoid is called genome. So you can say plasmid is what basically? We can also say it is extracircular DNA. It is extracircular DNA. Getting the things or not? Now, if DNA is there, they will do for job, some job, na? Because gene is there. And gene is there, so genotype is there due to gene. And if gene is there, they will do the some function. So they change the phenotype of the cell. Able to understand? Phenotype means what? Physical expression of the cell. So what are the job done by the plasmid? Function. See, it not doing any main function. Even in a cell, plasmid is not there, cell will survive. But when plasmid is there, it is doing some additional function. One function I am writing, antibiotic resistivity. Provide resistance to antibiotic. It have a gene who provide resistivity for antibiotic. So provide resistivity against antibiotics. It provides resistivity against antibiotic. Who is providing resistivity against antibiotics? Plasmid. Second thing, we use plasmid as vector. It provides resistivity to whom? Resistivity to bacteria. Don't confuse. Against antibiotic. So we use plasmid as vector in genetic engineering. GE I wrote in short. Third function, it used to identify the transformant during rDNA technology, recombinant DNA technology. Now, see, it is a long-term batch, so you can understand it well. In biotechnology, we study ampicillin gene, tetracycline resistance gene. So these are going to help us to differentiate between transformant, non-transformant, recombinant, non-recombinant. So I wrote these lines. NCERT also wrote this. So you got the idea, means use to identify. We will deal how to use to identify in which one? Biotechnology. Understanding the idea. So it used to identify the transformant during which one? or DNA technology. So this is about the plasmid. Much more about plasmid we will study in which one? Biotechnology. So after plasmid, we are going to talk about polyribosome, or it is called polysome, or it is called polysome. Understanding the idea? Now, what is this polyribosome? One mRNA and many ribosome associated with it. So how many mRNA? It is what basically? mRNA is there, and these structure are what basically? Ribosome. So examiner will ask sometime, in polyribosome or polysome, what is there? Many ribosome associated with which one? Single mRNA. That is called polysome or polyribosome. What is the function of polysome or polyribosome? Function is protein synthesis. Translation. Got the idea? So ribosome is the site of protein synthesis. So polysome, polyribosome is also over. Now, who remaining in the cytoplasm be left? Inclusion body. Inclusion bodies. Or body simply, right? Not a problem. Inclusion body. So, what is inclusion body? Inclusion bodies are non-membranous. Be very careful. Membranous or non-membranous? It is non-membranous. A storage granules. It is non-membranous what? A storage granules present in prokaryotic cell. So it is non-membranous storage granules present in prokaryotic cells is called inclusion body. And I am writing some examples of the inclusion body. First example I am writing phosphate granule. Second example I am writing glycogen granule. Third I am writing synophysian granules. And fourth I am writing gas vacuole. So phosphate granules store phosphate. No one will ask an exam. Very direct, na? Phosphate granule, anybody will ask? Store what? Phosphate? No one will ask. Glycogen granule store glycogen. Name is showing the thing. But here name is not showing. Synophysian granule, these examiner will ask. It is storing starch. So what storing what in inclusion body? This is the question going to ask. Everything named clear by the name. Gas vacuole store air. Now question arises. Here, these gas vacuole, you just try to understand. Don't confuse. Here tonoplast is not there. Here membrane is present, but it is not made up of phospholipid bilayer. And that is the reason, beside of presence in the membrane, we are presenting it along with a non-membranous structure. Because it is not a true membrane of phospholipid. Able to understand? And these gas vacuole is present in photosynthetic bacteria. Got the idea? Photosynthetic bacteria. Example, blue-green algae, BGA. Another example, iron, sulfur, and green sulfur bacteria. Now question arises, Why these gas vacuole present in these photosynthetic bacteria? So these gas vacuole provide buoyant forces, provide buoyancy. It provides buoyancy. So due to that reason, who will float on the surface of water? Bacteria will float on the surface of water, photosynthetic bacteria, and it got proper amount of light for which one? Photosynthesis. It got proper amount of light for which purpose? Photosynthesis. You got the idea? Understanding came? Now we are going to talk about flagella. Flagella. So it is a long filamentous extension arise from, and I am writing a star here, cell wall. It is written in NCERT. And why I am mentioning a star here? Because NCERT not wrote it correctly. So I am striking this word. Got the idea or not? So it is long filamentous extension arise from cell wall. Flagella, you use your common sense. Flagella can move. Locomotion. What is the function? Locomotion. So use your common sense. If anything arises from the dead layer, locomotion require energy, na? Try to understand. Locomotion require energy. Anything which is arising from dead air, how they provide the energy for locomotion? Understanding the idea? Clarity came in the mind or not? Right. So it is not correct to it arise only from which one? Cell wall. I am telling you, I am not going to say in the detail, but I am giving you a basic idea. You will understand. Look here. Suppose that this is a cell wall and this is the cell membrane. Cell membrane. Understanding the idea? It is, this layer is what basically? Cell membrane of bacteria. So what happened basically? Flagella have how many components? 3 components. Flagella have how many components? 3 components. One component is basal body. Basal body. Another component is hook. Another component is filament. So these basal body have 4 rings. These basal body part of the flagella embedded in which one? Cell wall and cell membrane both. With the help of how many ring? 4 ring. Out of 4 ring, 2 ring is present in cell membrane. And out of 4 ring, 2 ring is present in which one? Cell wall. So these are cell wall, cell membrane, inner, and these are outer which one? Cell wall. So basal body is partially, what is the correct thing? Basal body is partially embedded in which one? Cell wall. Partially embedded in which one? Cell membrane. Understanding the idea or not? It is not only embedded in cell wall, it is not correct to say. It is not only embedded in cell wall, it is partially embedded in cell wall. I am not telling you detailing of it. Getting the things? I don't want to confuse you unnecessarily, but beside of that, if it is coming in NEET, Kiji, flagella in prokaryotic cell arise from, answer cell wall only. What is the thumb rule? Even NCERT wrong, we have to follow it. What to do? In last, this is the most important tools you have now when any question came in controversy. And you have answer according to other books. An examiner give according to NCERT. Then usually they not accept your claim in controversy. Why? They will say, I am not taking guarantee of what written where in which book. This is at least government book, so you can say, you can claim ki bhai you have given the book. Government book is this. You are prescribing to read it. If you wrote wrong, what I can do? What you wrote, I am a student. I answered it. So in any controversy, what is the things to do? Follow the NCERT. Beside of that, it is wrong. But beside of that, it is our moral responsibility to say where what is wrong. Understanding the idea, picture came in mind or not? Right? Is it clear? So even it is coming in exam. Otherwise, student came with doubt. In eukaryotic cell, they are writing what? Plasma membrane. In prokaryotic cell, they wrote cell wall. Student came, ki sir, what to do about the flagella? That's the reason I have given this clarification to you people. I hope you understand it, what I want to convey. Is it clear? Is it all right? Done? Can we move ahead? So flagella have, so look now here, these are basal body, and then later on with basal body, who will join? Hook will join. So suppose that this is the hook. One side the hook is joining basal body, another side the hook will join with which one? Filament. So who is the longest part of the flagella? Filament. Filament. In the flagella is which one? Longest part. Understanding the idea or not? And if you talk about flagella, flagella is made up of a protein called flagellin. So flagella is made up of which protein? Flagellin protein. So you don't confuse. Eukaryotic flagella is made up of tubulin protein. Prokaryotic flagella is made up of flagellin protein. In prokaryotic flagella, 9+2 arrangement absent. Arrangement is absent. What is 9+2 arrangement? I will tell you in the eukaryotic flagella. That arrangement is absent in which one? Prokaryotic flagella. So you can say it is unistriated. So who is over? Flagella is over. Now we are going to talk about Fimbri and Pili. Understanding the idea? Now we are going to talk about Pili first. So Pili, it is a tubular extension. Got the idea? And it is tubular extension outward. It is tubular outward extension. Getting the idea or not? And this Pili is responsible for, it have role in formation of conjugation tube during bacterial conjugation. This line not written in NCERT. Bacterial conjugation. Bacterial conjugation is a type of sexual reproduction in bacteria, and during this conjugation process, two bacteria join together with the help of sex pili, and it is called conjugation bridge or tube. Conjugation tube, and through this tube, DNA from one bacteria moving into which one? Another bacteria. DNA of one bacteria moving into which one? Another bacteria. Understanding the idea? Picture came in the mind. Is it clear? Right? So this is about which one? Pili. These are the conjugation tube made up of which one? Pili. Understanding the idea or not? Clarity came in the mind. Now we are going to move ahead. Now we are going to move ahead about Fimbri. This Pili is also proteinaceous structure made up of Pilene protein. Made up of Pilene protein. Got the idea? Now I am telling Fimbri. Fimbri is what? Bristol-like extension present all over the body, and it is a small bristle like small extension present all over the body. And these fimbriae have a role in attachment or adherence, adhere the bacteria with rock and host cell. Now, when you read NCERT, NCERT author wrote the function of Pili and fimbriae together, and it looked like they wrote this function only. They not wrote separately about Pili and fimbriae. So be careful about it. Many times paper setter do ask a question like that. Ki ji Pili and fimbriae have role in adherence. If they are asking out of Pili and fimbriae, who have role in adherence, so then better answer what? Fimbriae. Because when you read the NCERT textbook, they not segregate the function of which one? Pili and fimbriae. Are you getting the idea? So it adhere. Adherence means what? Attachment. It helps to attach the bacteria with which one? Host cell. So who is over? Prokaryotic cell is over. Got the idea? Now we are moving toward eukaryotic cell. So should we continue, or you want 1 or 2 minutes of break or 5 minutes of break? You tell me. I have no problem. So before eukaryotic cell, you just want to take somehow drinking water, relaxation, whatever is it. Or should we continue? Someone writing break. Okay. Break you wish? Okay. Take the break of 2-3 minutes, then we will start once. Okay. Right. So I'm not going to once again end the streaming and start the streaming. It is continued. So you take the break of 1 or 2 minutes. I will just see you in 2 minutes. Okay, ji? So let's start once. We are going to start with eukaryotic cell. Right. Prokaryotic cell is over. We are going to talk about eukaryotic cell. Now, if we talk about eukaryotic cell, I already told in difference between prokaryote and eukaryote. There are some special structure present in eukaryotic cell is what? One is compartmentalization. I explained compartmentalization already. What is this compartmentalization? Cells is divided into small compartment due to presence of the cell organelle. In eukaryotic cell, DNA is organized into chromosome. In eukaryotic cell, cytoskeletal structure are present, which is not present in prokaryotic cell. And in eukaryotic cell, well-organized nucleus is present. Is it all right? That I already told. So if we talk about eukaryotic cell, so it present in protist, fungi, plantae, and animalia. So except Monera, all the four kingdom of which one? Whitaker have which kind of cell? Eukaryotic cell. Is it clear? Now we are going to talk in detail about eukaryotic cell. First, I am giving you a basic flow chart. So I am talking about a typical eukaryotic cell. Basic terminology you must know before moving. Typical eukaryotic cell have two components. One component is cell wall and another component is called protoplast. Protoplast means in eukaryotic cell, everything other than cell wall is called protoplast. Everything other than cell wall is called protoplast. So what is everything other than cell wall? What is present in protoplast? So protoplast have two components. One component is plasma membrane. And another component of the protoplast is protoplasm. So what is the mathematical language we can use? We can say protoplast equal to plasma membrane plus protoplasm. Protoplast equal to plasma membrane plus protoplasm. All together we call it protoplast. Now this protoplasm further have two components. One is what? Cytoplasm, and another component is nucleus. So you can say cytoplasm and nucleus together called protoplasm. If you remove nucleus, if you not talk about nucleus, the remaining part of the protoplasm is called cytoplasm. Now if we talk about cytoplasm, cytoplasm have three type of component. Cytoplasm have three type of component. One type of component is called cytosol. Cytosol. Another component is cell organelle. And another component is ergastric substances. Ergastric substances. Got the idea? So if we talk about cytosol, it is fluid filled matrix. Right. So what is cytosol? If you remove cell organelle and ergastric substances from the cytoplasm, what fluid component will remain in the cytoplasm is called cytosol. You got the meaning of cytosol? If we remove everything from everything means what? Cell organelle, ergastric substances, from the cytoplasm. What the fluid component removed is called cytosol. Now cell organelle, if you talk about the cell organelle, so we categorize this cell organelle in three categories. We categorize cell organelle in three categories. Category number one is DM, SM and NM. Double membrane bounded, single membrane bounded and non-membrane bounded organelle. So double membrane bounded organelles, you better know. What are the name of double membrane bounded organelle? It is plastid and it is mitochondria. Mitochondria. These two cytoplasmic organelles have double membrane. But nucleus also have double membrane, but it is out of cytoplasm. Getting the idea? Now single membrane bounded organelle is what? ER, Golgi body, lysosome and vacuole. These are single membrane bounded cell organelle present in cytoplasm, and non-membranous organelle is ribosome and another is centrosome. These centrosome contain two centriole. Are you getting the idea? Is it clear in the mind? Now ergastic substances. Ergastic substances are what basically? These are non-living substances. And these ergastic substances are a storage product present in the cell, excretory product present in cell, it is secretory product present in cell. These all are what basically? Ergastic substances. One example I am giving for understanding. A storage product, an example, in a cell starch is there. A starch is stored in the cell, na? Where it is stored? It is stored in cytoplasm itself. So it is a which kind of substances? Ergastic substances. I am telling tannin. Tannin is what basically? It is excretory product of the cell. Secretory product, pigments, are what basically? Or you can say nectars, or what basically? These are secretory product of the cell. So they come under which one? Ergastic substances. Now I am drawing a diagram of a typical cell. Look here. This is cell membrane or plasma membrane, and this is nucleus. So in between protoplasm, in between plasma membrane and nucleus, who is there? Cytoplasm is there. Cytoplasm plus nucleus together called which one? Protoplasm. When we add a plasma membrane in it, it is called protoplast. So a basic aspects of the cell came in the mind or not? Clear? Now we are going to start with which one? Cell wall. What we are going to start? Cell wall. Give your attention. Cell wall. So it is outermost covering in some protistans fungi and plantae. In eukaryotic organisms, cell wall is absent in animalia. So out of 4 kingdoms of eukaryote, cell wall present in member of 3 kingdoms. And why I wrote some protist, there is a reason. Some protists don't have cell wall, like protozoa and protist don't have cell wall. Slime mold don't have cell wall. Euglenoid don't have cell wall. Cell wall present in which one? Diatoms, that is chrysophytes and dinoflagellate. Understanding the idea or not? So these are the occurrence of cell wall, where cell wall is present. Picture came in the mind. After that, I am telling about composition of cell wall. Chemical composition of cell wall. Chemical composition of cell wall. So what we report in the chemical composition of cell wall? In algae, cell wall is made up of cellulose, galactans, mannans, and mineral like calcium carbonate. So this is the composition of algal cell wall. Getting the idea? Now I am telling in higher plant, what is the composition of the cell wall? It is cellulose plus hemicellulose plus pectin plus protein. So these are the chemical composition of cell wall of higher plant. Understanding the idea? Is it clear in the mind? Now I am asking a difficult question. Try to answer it. If I ask a question, is hemicellulose is present in algal cell wall, then what will be your answer? Tell me, hemicellulose is present in algal cell wall or not? Is hemicellulose present in algal cell wall? So two type of answer came from your side. One is no, another is yes. You have studied anywhere about hemicellulose written in algal cell wall? It is written in algal cell wall. NCERT not told about ki hemicellulose either presence or absence in algal cell wall. NCERT said only about hemicellulose is what? Present in higher plant cell wall. It is written in NCERT, hemicellulose is absent in algal cell wall. Written or not written? Not written, na? Not written, na? So why you are assuming not written means not present? If anywhere any information is not written, it doesn't mean what? It is not present. So it is called misinterpretation. Certain time a student, certain time teacher make this misinterpretation. Ki not present. How people interpret it? Hemicellulose written present in higher plant, not written in algae. So many times people misinterpret it. It means hemicellulose is absent in algal cell wall. It is not true. It is not true. Hemicellulose is also present in which one? Algal cell wall. But it is not written in NCERT. Information is not given. So not written is doesn't means we will say absent. Where it is written, if not written means absent. Understanding the idea? So what message I want to give, it is very crystal clear in your mind or not? And that is the reason I told it is a difficult question. Because your textbook is silent about it. They not wrote about either it is present in algae or not. That clarity came in the mind. Can we move ahead? Understanding came in mind? Is it? Yeah. Very good. Now give your attention. Now, I am going to tell you about the cell wall. So, just try to understand. Somehow, I just want to deal conceptually. Because you know, I know about what line is written in NCERT, you will know. But I am just telling you the concept behind it. Look here. How the cell wall formation occur. Getting the idea? Not much detail I will tell, but I am going to tell you at to certain level of the explanation. Not much explanation I am giving. So look here. So when a cell divide, first what happened? Karyokinesis occur. Nuclear division occur. Suppose that this is a plant cell where karyokinesis is completed. Who divided? Nucleus divided. Nuclear division is over. Now a question arises, how cytokinesis occur? Now a question arises, how cytokinesis occur? Is it? One student asking difference between hemicellulose and cellulose. So you will study in biomolecule, but you ask the question, I am answering. Cellulose is a polymer of glucose, homopolymer, and in hemicellulose it is a heteropolymer. So many different another structures are there. Got the idea? So what I am telling, look here, this is the nucleus. Now problem is what? In cell wall formation, do you know what is the problem in cell wall formation? Problem in cell wall formation is endoplasmic reticulum. You try to understand the mechanism. These endoplasmic reticulum you have studied, now it is a network like reticular structure, and these reticular present in the entire cytoplasm. So when in entire cytoplasm endoplasmic reticulum is present, then a question arises then how the formation of cell plate occur or cell wall occur? Everywhere endoplasmic reticulum is there. Who is there? Endoplasmic reticulum is spread in everywhere in the cell. Endoplasmic reticulum is present everywhere in the cell. Now question arises, this is a plant cell and here cell wall formation required. How cell wall formation occur? This pipe of endoplasmic reticulum create a problem or not? This tubule of the endoplasmic reticulum creating a problem or not? I am somehow making a narrower tubule. Right now, this will create a problem or not? Tell me yes or no. This will create a problem. So how the cell wall will form? Let me tell you. So first of all, what happened? Formation of phragmoplast occur. How? I am telling you, you will understand the mechanism. Some patience required for understanding. So microfilament, I am telling you, microfilament, microtubule, and endoplasmic reticulum, ER, all together form phragmoplast. Now these phragmoplasts help in formation of cell plate. Help in formation of cell plate. So who is forming the phragmoplast? Microfilament, microtubule and ER, endoplasmic reticulum forming phragmoplast. And these phragmoplasts help in formation of which one? Cell plate. During formation of cell plate, what happened? Vesicles of Golgi body assemble, and these assemblies start from center and move in periphery. They are assembling like this. Who is this? Who is assembled here? Golgian vesicle assemble, and their assembly occurs centrifugally, centrifugal direction from center to periphery. And this structure is what basically cell plate. So during cell plate formation, who help the assembling the which one? Golgian vesicle. Here it is Golgian vesicle. So it is the vesicles formed by the Golgi body, but who helping the arranging these particular Golginian vesicle in such a way? These phragmoplast helping to arrange the Golginian vesicle in such a way, and then who is forming? Cell plate is forming. Got the idea? So before cell plate formation, who get assembled? Phragmoplast get assembled. These phragmoplast utilizing which one? Helping which one? Golginian vesicle to arrange like this. And this structure is called cell plate. Later on, this cell plate converted into middle lamella. Middle lamella. So NCERT wrote, Bhaiya, between two cell, middle lamella is present. Suppose that between two cell, later on, these phragmoplast converted into which one? Middle lamella. So this is the middle lamella. So you got the picture behind the middle lamella, how these middle lamella form, understanding the idea, picture clarity came or not, tell me honestly, middle lamella, middle lamella. So for me it is easy to say middle lamella present. Less energy required, less time required, but beside of that I am telling, it's not like it not have a significance. You must be conceptually smart. And these are the topic also going to use further. Getting the idea? So that is the reason I am investing time in this particular area. What I am telling, here it is who is there? Microfilament is microtubule, ER, all together forming the phragmoplast. These phragmoplast help in assembly of which one? Golginian vesicle. When Golginian vesicle assemble, they are forming which one? Cell plate. And cell plate later on converted into middle lamella. Entire picture came or not came? Tell me very honestly. Tell me very honestly. Understanding came? How the middle lamella form? Is it? Now I am telling about this middle lamella. This middle lamella is cementing material between two adjacent cells. Two adjacent cells. And it is made up of calcium and magnesium pictate. So now this is cell A and this is cell B. Now who are present between these two cells? Middle lamella is present between these two cells. These are the cell cement. These are the cementing material between the two cells. Picture came in the mind. Is it all right? But where middle lamella is not there? Where tubes of endoplasmic reticulum is there? How they can pass the tube? Tube is intervening, na? They are not giving the free space. So overall, who can form? Middle lamella going to form. So these middle lamella is not present where these tubes of ER is there. Understanding the idea? Now, this tubule of the ER who are present is called, I am writing the name desmotubule. It is called what basically? Desmotubule. Are you understanding the things or not? Later on, inner side of the, which one? Later on, inner side of the middle lamella, both side, formation of primary cell wall occur. Primary cell wall form. Here also primary cell wall, primary cell wall. Getting the thing? So middle lamella is common between two cells. Inner side of the middle lamella who is forming primary cell wall. Later on, when cell mature, layers of, three layer of secondary cell wall form. S1, S2, S3, S1, S2, S3, S1, it is S1, S2, S3, S1, S2, S3. Getting the thing? So I am writing here primary wall. I am changing the color. Look here. It is primary cell wall, green color. And these are what? S1, S2, S3, secondary cell wall. Able to understand? So in such a way, formation of the which one occur? Cell wall occur. And now I am drawing the plasma membrane. You will understand it better. Now here you look. Inside of the cell wall, everywhere, who is present? Plasma membrane is there. Who is there? Plasma membrane is there. Now I am asking a question to you. And here who is there? Cytoplasm is there. Cytoplasm is there. And these blue color tube which desmotibules are there. So are you able to understand here that these two cell, here it is, in these two cell plasma membrane is discontinuous or continuous? Cytoplasm is continuous or discontinuous? One student asking, sir, will this class go on till NEET 2024 on YouTube? No. Few more days it is on YouTube, then it will shifted totally on the app. Right? Or app of Sri Gosalite. Now, are you able to understand? Clarity is coming in the mind. So these two cells are interconnected with which one? Plasma membrane, na? Continuous with plasma membrane. Na? Getting the things or not? A cell and B cell. And what is this structure where cell wall is not there, where middle lamella is not there? This structure is called plasmodesmata. Plasmodesmata is most ignored thing. NCERT also ignored it, right to one line. Plasmodesmata. What is plasmodesmata? Let me tell you about plasmodesmata. Plasmodesmata. So it is microscopic pore between adjacent cell. Between two adjacent cells, microscopic pore is called plasmodesmata. This is the microscopic pore present between which one? Two adjacent cells. NCERT wrote what? Plasmodesmata transverse the cell wall and middle lamella. You are finding that through the cell wall and middle lamella a pipe is passing from one cell to another cell. Getting the things or not? And cell wall is not there. Getting the thing? So wherever pipe is there, cell wall material not deposited, and even formation of who occur? Pits occur. Because wherever desmotibule is passing, in that area cell wall material not form, and there is a formation of who occur? Pits occur. And through that a continuous pore present between two cells. So when you read plant physiology in NCERT, there is a line written. You will got the understanding of that line here only. Try to understand. When you read NCERT, NCERT wrote, symplast is a pathway of interconnected protoplast. Two cell protoplast is interconnected or not? Through what it is interconnected? Plasmodesmata it is interconnected. So now you just look, I am amplifying this diagram. Just give your attention. I am amplifying this diagram. I am making this diagram bigger. You just try to understand here. There is a gap present between desmotibule and plasma membrane. Tell me, look at the diagram and tell me. This is red color, pink color line is plasma membrane. Blue one is desmotibule, and here it is a gap. Here it is a gap. This area is called what basically? Cytoplasmic sleeve. Cytoplasmic sleeve. Got the meaning? Cytoplasmic sleeve present between plasma membrane and desmotibule. Desmotibule is a modified endoplasmic reticulum. Understanding the idea or not? Getting the idea or not? So plasmodesmata made up of how many components? Plasmodesmata is made up of three components. One component is plasma membrane. Plasma membrane, you have to know, it is most ignored area. And another thing is which one? Cytoplasmic sleeve. And it is very important area. Cytoplasmic sleeve. And third component is desmotobule. And what is the function of this plasmodesmata? Function of this plasmodesmata is exchange of material between adjacent cell. So plasmodesmata helping the transport of material from one cell to another cell. Suppose that something required in this cell, that thing move through plasmodesmata from here to here. Something required in this cell, that is moving through plasmodesmata from this cell to this cell. Even two cells connected together with the help of their ER. ER of this cell and this cell are continuous. How they are continuous? With the help of these desmotibules. Understanding the idea? So now I am going to tell you about the basics of the cell wall. That is what? Cell wall. Middle lamella I already told. So cell wall, I am telling about primary cell wall, secondary cell wall, and tertiary cell wall, two line, and tertiary cell wall. Tertiary cell wall. If we talk about primary cell wall, this primary cell wall is thin and elastic, present in all plant cell. Secondary cell wall is thick and rigid. Thick and rigid, absent in meristematic cell. Absent in meristematic cell. Got the idea? And after formation of secondary cell wall, primary cell wall diminished. Primary cell wall diminished. What is the meaning of this line? Let me tell you with the help of example. Let me tell you with the help of a beautiful example. I have drawn here a line. Are you able to see it? Are you able to see it? Yes or no? Yes. It is primary cell wall. It is thin and elastic. Now I am drawing another line of different color. I am drawing here. Try to understand. I am drawing another line. First line is looking you clearly. Not na? So when a thicker layer came near to which one? Thinner layer get diminished. That is written in NCERT, and many times students struggle getting the thing. So what is the meaning of this, sir? Meaning came or not came? Not clearly visible, na? And that is the reason NCERT wrote कि when secondary cell wall form, who diminished? Primary cell wall get diminished. Understanding the idea? Clarity came or not? And now I am writing one line about tertiary cell wall. Where it is present? It is present in, not everywhere, it is present in tracheids of gymnosperm. So I can finish cell wall in 10 minutes even, but why I told much about cell wall? To make you understand the plasmodesmata. Otherwise people do not understand plasmodesmata. They pass the NEET certain time if question not came, and even don't have idea much about which one, plasmodesmata. Getting the idea or not? So every time you must be very analytical. Every time not coming question directly coming from the line. You must be ready for the all situation. Either paper is easy or difficult, I have to handle. You cannot take at a granted this year only line came, sir. Last year people struggle in the botany. So you must be very perfect in your concept. Understanding the idea? So now I am telling you about the function of cell wall. Then who will over? Cell wall will over. Function of cell wall. And you tell me the function of cell wall. Why should every time I say? So it is what? Protective in function. Provide mechanical support. Mechanical support to cell. Provide shape to cell. It is responsible for cell to cell interaction. NCERT wrote this line. Cell to cell interaction. Getting the things. It acts as barrier for macromolecules. And if someone asks cell wall is a permeable or selectively permeable or semi-permeable? Permeable structure. If some more thing deposit on the cell wall, it become impermeable, like sovereign, like lignin, then it become permeable. Normal cell wall are what basically? Permeable. One student asking difference between pit and plasmodesmata. Pit is a depression in the cell wall, simply. Pit is a depression in the cell wall, and usually this depression present in the area where plasmodesmata is there. Are you getting the idea? And plasmodesmata consists of desmotubule, plasma membrane and cytoplasmic sleeve. So these plasmodesmata performing the exchange between two cells. Exchange of material between two cells. Pit is simply a depression in the cell wall, but where this depression is present? Wherever plasmodesmata is there. So understanding came, what is the difference between pits and plasmodesmata? Wherever desmotubule is there, cell wall not formed. Clear? Now we are going to start with a new topic, that is what? Cell membrane. Plasma membrane. So let's start with the plasma membrane. So it is thin and elastic. It is universally present. Not any single cell is there where who is not there? Plasma membrane is not there. It is universally present in every cell. Plant cell, animal cell, prokaryotic cell, eukaryotic cell. It is selectively permeable. Selectively permeable membrane. And if we talk about, it is a leaving membrane. It is a leaving boundary. Plasma membrane is also called which one? Leaving boundary. Now we are going to talk about the chemical composition of plasma membrane. Chemical composition. So if we talk about chemical composition of the plasma membrane, it is made up of lipid, also protein, also carbohydrate. So lipid plus protein plus carbohydrate, all three are present in the plasma membrane. If we talk about which type of lipid is present in plasma membrane, so I am writing few important names. Phospholipid, another is cholesterol, another one is glycolipid. So these three type of lipid is present in plasma membrane. If we talk about protein type present in plasma membrane, so the protein who is present in the plasma membrane is of two types. One is extrinsic protein, another one is intrinsic protein. Is it clear in the mind? If we talk about extrinsic protein, it is also called peripheral protein. And intrinsic protein is also called integral protein. And these protein is classified based on ease of extraction. So the protein who are easily extracted from plasma membrane is called extrinsic protein, and who are not easily extracted is called intrinsic protein. Now this intrinsic protein is of two types. Partially buried protein and totally buried protein. Partially buried protein or totally buried protein. I will show the diagram, you will understand better that time. Totally buried protein have another name, it is also called transmembrane protein. Transmembrane protein. Getting the idea or not? An example of this transmembrane protein is carrier protein, channel protein, tunnel protein, pump protein, carrier or channel or tunnel, same thing, pump protein. These are the example. I will say about all, don't worry about it. So I classified the protein into how many category? Two category. One is what? Extrinsic, another is intrinsic. Extrinsic is also called peripheral, intrinsic is also called integral. Integral protein is of two types, partially buried and totally buried in the membrane. Who are totally buried is called transmembrane protein, example of transmembrane protein. Understanding the idea or not? Clarity came in the mind. Now I am just writing the percentage of protein and lipid in a plasma membrane is variable from membrane to membrane. Membrane to membrane. It is not a fixed percentage is there. What NCERT wrote? NCERT wrote in human RBC. In human RBC, 52% protein is there in the plasma membrane and 40% who is there? Lipid is there. But it is not a constant percentage. These percentage is variable from plasma membrane to which one? Plasma membrane. Understanding the idea? These percentage is variable from plasma membrane to plasma membrane. Understanding the idea? Picture came in the mind or not? So what are the chemical composition of plasma membrane is there? You got the idea? It is made up of lipid, it is made up of protein, it is made up of carbohydrate. And how many lipid I told? Three type of lipid I told. More is also there, but it is not at your standard. One is what? Phospholipid, cholesterol, and glycolipid. Let me tell you about a basic idea about the phospholipid. Phospholipid. So if you talk about a phospholipid, these two component is present in a phospholipid. I will tell you the structure of phospholipid in which chapter? Biomolecule. So one component of phospholipid is what? Basically head. And another component of phospholipid is tail. So this tail is made up of fatty acid. Tail is made up of fatty acid. Here two tail is there. So R1 and R2 I am writing. Two fatty acid is there. And head is made up of two component. One is what? Glycerol. And who fuse with the head? Phosphoric acid. And that is the reason here I am telling two tail only. So this head part of the phospholipid is hydrophilic. And tail part of the phospholipid is hydrophobic. It is what? Basically hydrophobic. Head part is hydrophilic and tail part of the phospholipid is what? Basically hydrophobic. Fatty acid chain is there. Understanding the idea or not? Who is there? Fatty acid chain is there. So these phospholipid present in plasma membrane as bilayer, two layer of phospholipid, and it is called which one? Phospholipid bilayer. So wherever you see the plasma membrane, you will find phospholipid present like this. And these phospholipid form a bilayer, and that is called which one? Phospholipid bilayer. So this is what basically? Phospholipid bilayer. Phospholipid bilayer. So who is always present as a bilayer? Because they have to protect their tail. So tail is hydrophobic, it is away from the water, and here who is there? Water is there, here water is there. They are keeping their tail away from the water, so they are shifting their head toward the water. Getting the idea? And that is the reason it is present as phospholipid bilayer. If I give you phospholipid, and ask you throw in water, do you know what will form? Do you know what will form when you throw the water, phospholipid in water? Formation of a micelle occur. After the class, Google the structure of a micelle. It arranged in a bilayer. Understanding the idea or not? So when you throw the phospholipid, so when micelle formation occur, two environment created, na? One outer environment, another inner environment in a micelle. So both side head. It is like this, and center who is there? Tail is there. Understanding the idea? So when a micelle formation occur, both toward inner and outer side who is there? Head is there. So this is the cytoplasm form, and this is outside, and this is phospholipid bilayer. This is the phospholipid bilayer of the cell membrane. So formation of a micelle occur when you throw the which one? Phospholipid into the water. Understanding the idea? Picture came or not came? So you got the idea. What is the reason behind phospholipid present as a bilayer? Because tail want to protect itself from the water. So they are placing the head toward the water. So two tail are just away the water. Suppose that when I want to save my back from water, So what I will do? I will take the hide myself with another person. So we are just going to join by back to back, because I don't want to raining occur. And suppose that I want to save my back. So two people arrange with the back. So both back will save. Who is going to face the water? Front is going to face the water. Are you understanding the idea? Picture came or not came in the mind. So up to here it is okay or not. Now I am going to tell about time is there or time up. Time is there or time up? Up to time over. Next class is which subject? LC sir next. Okay. So in next class we will discuss about fluid mosaic model. Which one? Fluid mosaic model of the plasma membrane. Okay ji, thank you very much for your patience for listening. Bye bye.